Welcome, everyone. This is Christine Buckley with the Brain Aneurysm Foundation, and happy you're joining our monthly webinar series. And we have a pretty interesting topic this month, and we are super happy to have Dr. Christopher Jackson from neurosurgery at Johns Hopkins join us to talk about the research progress in vasospasm and delayed cerebral ischemia, which we will call DCI moving forward, just to keep things a little more simplified. So Dr. Jackson, would you like to say a few words before we get going?
Of course, yeah. Thanks for the invitation, Christine, and thanks to everyone who can join. Yeah, so as Christine mentioned, I'm a neurosurgeon here at Hopkins and a researcher. I have a particular interest in inflammation and how inflammatory processes and sort of poor regulation of the immune system drives some of the outcomes after aneurysm rupture and in other cerebrovascular diseases. So look forward to talking a little bit about that with you guys this afternoon, and hopefully also answering some more important, sort of fundamental questions about vasospasm and delayed cerebral ischemia and what it means and why we care about it and where we need to go. So thanks for the invitation. Thanks for joining.
All right. Thank you, Dr. Jackson. So to get started, do you mind defining vasospasm and DCI, including at what point these conditions might impact a patient in their brain aneurysm recovery?
Yeah, absolutely. So this happens in sort of the subacute period. And so I think, when I talk to my residents, and this is the same way I explain it to patients, you have to think about after an aneurysm ruptures, what the risks are at each point in time, right? So if an aneurysm ruptures, and the immediate risk is that you have bleeding into the brain, right, under high pressure. And that bleeding needs to stop, and then the person needs to be stabilized, right? So the first thing that we do is we make sure that their airway is protected if they're unable to protect their own airway, that their heart is functioning properly, the basic things, to get somebody supported so that we can move on to saving their brain, right? The next thing that we have to do is secure the aneurysm. And you guys have heard probably a lot about that from others, many techniques, endovascular, open surgery, and that needs to happen within 24 hours. So that's sort of the rush up front. And that's where a lot of the focus has been, and even in research. Vasospasm and delayed cerebral ischemia are what happen 3 to 14 days after the aneurysm ruptures. So, it almost never occurs immediately. In some patients, particularly young women, and we'll get a little bit to why that might be. I think it's because of immune responses that are more robust in that population. You can see it at 48 hours, but typically it can start at 72 hours. It peaks around day 8 and interestingly stops on its own at day 14. And so, so that's sort of the time period. And in terms of what it is, so it's important to distinguish vasospasm and delayed cerebral ischemia because they're connected, but they're not necessarily the same thing. So vasospasm is a process by which the arteries in the brain are irritated because of the blood and they pinch off, right? And so any of you who've either unfortunately gone through a ruptured aneurysm or a loved one who's had a ruptured aneurysm knows that every day somebody would come in with an ultrasound and insonate different parts of the head to look for what transcranial Doppler ultrasound. So what that's looking at is how fast blood is flowing through the vessels. It's just like when you're a kid and you take a garden hose and you pinch the hose a little bit so that the water shoots out further and faster, right? So that's what that's doing. It's measuring how fast the blood is going through the vessels as a way of understanding or approximating how pinched off those vessels are. So that's vasospasm, and that we believe is an inflammatory process, but that's due to the blood that's around the brain. And in fact, if you look at patients most at risk, it tends to be patients who have more blood around the brain that fits with that. Delayed cerebral ischemia is what happens when you can't compensate essentially for the vasospasm. And what I mean by that is as the vessels pinch off, blood is not routed correctly, throughout the brain. But we have lots and lots of what we call collaterals, or all the arteries are connected in the brain. So, if you pinch off one artery over here, it may very well be in an individual that you have a pathway to get blood another route. So, I tell patients that's very similar to say, like, we're here in Baltimore. If you shut down Interstate 95, depends on what other roads are open. And often you don't know which other roads are open until you test that, right? Until somebody has to rely on those other pathways. When that is inadequate, then you get areas of the brain that don't have enough blood, just like a stroke. And that's what happens. And that's delayed cerebral ischemia, ischemia meaning a stroke. And so, it really is a function in a large part of the vasospasm that is stressing those collateral pathways, trying to get, and then if those collateral pathways can't keep up, you get this clinical event, which is basically small strokes. And it turns out that furthermore, that sort of causes, and this is where a lot of research is being done, sort of a downstream sequence of events where you start to get clots forming in small vessels and things like that that just further propagate the issue. But fundamentally, it's about pinching off the arteries days 3 to 14 and then not being able to get the blood there through other routes that get you to delayed cerebral ischemia.
Okay, thank you for that answer. That was great. Can you tell us what is the current treatment for these conditions and what are the biggest concerns with both vasospasm and DCI?
Yeah, so in terms of concerns, one is that we don't have a good way to predict who's going to get it, right? So we know that, the amount of blood in the head is a predictor. We know, again, in some patient populations, like I mentioned earlier, young women, that's a predictor, but it really can kind of come out of nowhere, and if any of you have been or have had loved ones who had an aneurysm rupture, but then, clinically did well initially, only to have delayed cerebral ischemia later, that, we keep patients in the ICU here for the entirety of that vasospasm period. So for 14 days, and that's whether or not the person, is severely injured neurologically or is up reading newspaper. And the reason for that is because we have no good way of predicting when it's going to happen. So that's one of the major issues and an area of research that many folks are working on. The other, and the concern too is that, after the initial aneurysm rupture, right, and then when the aneurysm has been secured, this is the #1 cause of delayed morbidity and mortality. So, this is what hurts people who, manage to do well through that initial event. And it's a little bit of sort of a wasteland, so to speak, in terms of research and options, right? So the only approved drug for vasospasm and delayed cerebral ischemia is nimodipine, which was approved based on a study in the 80s. And we haven't really had anything since, despite, lots and lots of efforts. So it's just supportive care. So it's fluids, it's making sure the blood pressure is kept up, which helps force the blood through those other collateral pathways, and intensive monitoring, because if patients do go into delayed cerebral ischemia and start to have strokes, then there are invasive maneuvers that can be done to reopen those vessels. So going to the angiography suite, or from inside the vessel, vasodilating, or in other words, medications that open the arteries can be injected, or sometimes even a balloon can be placed to mechanically, force the artery open. So, that's kind of what we're dealing with when we think clinically about vasospasm DCI.
Okay, thank you. Can you tell people about your BAF funded research project and what that funding has led you to move forward with?
Yeah, no, absolutely. So I have been interested. So I came at this from a slightly different way than some cerebrovascular neurosurgeons. So, my background was primarily in immunology and in tumor immunology before I sort of clinically became interested in aneurysms. In the world of tumors, there has been a lot of effort to target these anti-inflammatory pathways called checkpoints. And so you've probably seen commercials for Opdivo, Keytruda, for lung cancer, melanoma, et cetera. These are major advancements in oncology. And what a checkpoint is it's basically an off signal. So it turns out that immune cells express these signals that they say, okay, I'm going to go cause an inflammatory response unless you turn this signal on. And if you do, I won't. And, we have evolved to have those signals to prevent autoimmune disease. So those signals are used all the time to keep people from having autoimmune disease. Turns out cancer uses that against us. And so lots of efforts have been directed at turning those signals off so that the immune system can fight cancer. So I had this interesting idea years ago that, not too many years ago, but years ago, that maybe we could turn those signals on as an anti-inflammatory, and that maybe that plays a role in brain ischemia. It's sort of an out-of-the-box idea, which is why the Brain Aneurysm Foundation actually matters so much in my personal journey with this, because, a lot of people were kind of like, show me some preliminary data. The problem with that, of course, is that, you have to have preliminary data to get funding. You have to have funding to get data. This is the circle. and it requires sort of a trusted partner. And so, the Brain Aneurysm Foundation, with, our very earliest preliminary data, took an interest in this, and I was fortunate enough to get a grant to fund it. And as part of that, we've really been able to establish that immune checkpoints and the PD-1 immune checkpoint in general, specifically, can be turned on, at least in animals, to prevent vasospasm dramatically. In a specific kind of way where we're targeting immune cells in the periphery. So we're not trying to target the brain, but we're turning them off before they get there. But none of that would have been possible without the Brain Aneurysm Foundation, and then that is now developing. In my academic role here at Hopkins, I'm understanding what that is doing in terms of ischemia, because that's what we really care about, right? Is the delayed cerebral ischemia and the strokes. The vasospasm is one thing, but we think it's actually the strokes that are occurring because of it that are sort of creating this cycle of inflammation. So we're uncovering the mechanisms by how that happens and identifying drugs that maybe can be used, novel drugs, to turn those pathways on to prevent it in the first place.
Excellent. Please tell us about Egret Therapeutics and what they hope to accomplish regarding brain aneurysm patients.
Yeah, so Christine, I know you had an opportunity to meet the folks at Egret recently. So, and for full disclosure, I helped found that company and have stock in that company and all that. So take that with a bit of a grain of salt. But what I'll tell you is that they've been wonderful partners on developing this idea. So Egret Therapeutics was founded by myself and Dr. Michael Lim, who's the chairman at Stanford. And a couple of other people with a real passion for helping in this space, and they have identified a molecule that is a PD-1 agonist. In other words, it turns PD-1 on. So it turns out that that's hard to find. So there are lots of molecules out there, cancer drugs, to turn it off, but there are very few ways to turn it on. And they have identified and licensed a molecule to do that was being used by another company for basically organ transplantation. But when we approached them about using it for brain aneurysms, they were like, that's a small group of patients. That's, all the things that we often hear, from big companies. And but Egret Therapeutics was able to help license that. And we've been working with them to bring that drug to clinical trials to see if it can help patients with brain aneurysms that rupture, prevent them from having delayed cerebral ischemia and the effects of that. So it's exciting. It's been an exciting partnership, and they're making good progress.
Yeah, it is very exciting because, I mean, I know, again, through the number of people that we talk to, the problems that vasospasms and DCI cause people. So it is very exciting. And it's fine. You have stock and an interest in all this. It's just nice to know that you do have a partner to help carry this work through, because, I mean, we do know, again, the foundation is the largest private funder of brain aneurysm research. And, we are fortunate to be able to give someone like you a start. But I just know, as you do, for the people that come together through our symposium and the different discussions and seeing what people do, yeah, it's really hard to get the proper funding, but it's amazing what people do with the smaller grants that we do provide. So I think it is really amazing. So that's great to hear.
And to that point, Christine, as there's this, what we call in research, the valley of death, where, you discover something, which the Brain Aneurysm Foundation provides, a fantastic start for that, right? And even, the NIH and other funding sources can provide a fantastic start for that. But then all of a sudden, you have this finding and you want to get it into patients, and the funds required to do that, frankly, are far beyond what foundations and even the government often can provide. And so then, what is provided by the government is often for patients with common diseases, as you can imagine. So there are rare disease pathways and that sort of thing, but it's a minority. And one of the things that brain aneurysms have suffered from in terms of development, I mentioned that nimodipine is the only drug that's been approved for vasospasm, delayed cerebral ischemia, but is that some company basically has to take that leap, right? Some company has to partner with us, and take that leap and help us push it into patients and sort of bridge that valley of death. And I think, actually, I think it's critical that we partner with the people that are passionate about that. And, because that's the only way we're going to get drugs to patients. And it's long overdue for this indication. Sure.
Yeah. Thank you. And are there other people who are doing similar research that you're doing? And is there collaboration with different centers on this type of research?
Yeah, absolutely. I think we're taking kind of a unique perspective on this, again, sort of given, my background and interest in that we're looking at the immune cells before they ever get to the brain. So, I tell people that part of the thing that makes this so difficult to study is that, you can think about, a series of dominoes lined up that end up going all different directions. And often I think by the time you look in the brain, you're way down some pathway, where many dominoes have already fallen and it's hard to reverse. What's interesting about our approach is that we're looking at the immune cells in the blood that will eventually go to the brain, but we're sort of reprogramming them before they get there, right? And that's a very different kind of approach. So that part of our approach is unique, but many other people are looking at, sort of the specific damaging pathways downstream. And so neuroprotection is a big thing. How do we keep, neurons from dying, in the setting of when there is delayed cerebral ischemia? Looking at microthrombi, meaning very small clots in vessels that we can't see, and preventing that. A lot of folks are making good progress in that area, and trying to figure out, how we can, prevent that from happening. And then, even downstream of that, I think one area that gets lost a little bit in this whole discussion is, I won't say where or who, but I actually was giving a talk at a Grand Rounds once a few years ago, and was talking about vasospasm cerebral ischemia, and some of the audience said to me, that's not really an issue anymore with endovascular therapy because we can just open the vessel. But the problem is, it's, in my opinion, because we're not measuring the right things, right? So if you ask a patient, hey, can you hold your hands up? Yeah, they can hold their hands up. Can you carry on a conversation, enough to find a bathroom at a sporting event or something? Yeah, sure they can. But if you ask their family, they say they're different. Or if you ask them, they say, yeah, I've just not been able to go back to work the way that I did before. Or, recently actually I had a young patient who, interesting because his aneurysm was nowhere near, the parts of the brain associated with smell, actually sort of in a completely different zip code, if you want to think of it that way. But, he made a remarkable recovery from a ruptured aneurysm, but he can't smell anything and he can't taste anything. And he's 30 some years old, and he's going to be like that probably the rest of his life. But according to every metric that we commonly use to measure recovery, he's, an A+ success story, right? So I think it's but there are multiple efforts on all of those fronts. So this is certainly just complementary, and I think sort of in that chain of dominoes, I like to think that we're very early, but we're partnering with people who have experience in all those other events later, all the way from what happens, we're looking in the peripheral blood, but all the way to what happens in the brain, and then all the way to what happens to patients, months and years down the line. Because any intervention we make, we need to make sure, that the results follow all the way through and the patients are actually getting better, not just that we're making a picture better, an angiogram or an MRI.
Okay, thank you for that. What do you think is the next breakthrough in brain aneurysm research? What do you think that will be, and will it be for detection, treatment, or recovery?
Kind of similar to my last comment, I think actually all the above, and it has to happen in parallel. I think it's happening at a different pace in each of those areas. So, when you think about treatment of brain aneurysms, I mean, that is a constant march of technological advancements, and that's only going to continue to happen. So I think, I'm an open vascular neurosurgeon. I don't do endovascular. I do open and skull base. But, I think the endovascular technologies are coming fast and furious, and I think they will continue to. And I think, but for most aneurysms, actually, I think, patients are the better for it. We have to study those things, and make sure that they are in fact doing what they think that we're doing, that we think they're doing, and that they're durable and all those things. But I think that will continue. And I don't think we're likely to see sort of giant leaps in that area as much as continual progress. Some of the other areas you mentioned, like detection, there are several groups. I know some groups funded by the Brain Aneurysm Foundation, for example, the group at Buffalo, is looking at detection, looking at, basically blood tests that might be able to tell you, if you have an aneurysm, which would be a tremendous advance. I think, if those things come to fruition, the challenge there will be what we do with that information, right? So, do you screen everyone? that would be great. But then you also have this problem that you're going to have a lot of patients with a brain aneurysm that's not going to rupture, and there is some risk to treatment. So the next step from that is figuring out, which aneurysms are dangerous and which ones are not. There are lots of variables that we look at, but we're still not great at predicting that. There are some imaging methods, MRI-based methods looking at inflammation, again, near and dear to me, in the aneurysm wall, and other methods to try to determine which ones are going to be dangerous. But those efforts have to be complementary, because if you just go diagnose a whole bunch of aneurysms, then again, the vast majority of those people, actually, the aneurysm won't rupture. And you don't want to treat everybody, because then you're exposing them to the risk. So I think, those things are all happening in parallel. I think that the area of vasospasm, delayed cerebral ischemia, and sort of what I'll call, early subacute recovery is at the moment an area where we really just don't have a lot of options. I'm hopeful that what we're doing and complementary with what other people are doing will help to bridge that gap, because I think that's a critical period, kind of when the flurry of activity of taking care of somebody with a ruptured aneurysm is over. What do you do now? Because even though the patient looks peaceful and they're stable, we know biologically a lot is going on that's going to shape their outcomes. So I think that's a place that's ripe for sort of breakthroughs, because there's not been that continual progress. What we really need a paradigm shift, I think, for that. And then the other thing I already mentioned is long-term recovery. Like you said, how do we better train the brain to recover? How do we better understand what the deficits are and what the impact is on people's lives? And I think that is something that a lot of folks are working on, but is obviously a complicated problem. And it's honestly a partnership with patients, right? Like, unlike everything else we're talking about, where you can measure it, in a mouse or whatever, when you start talking about quality of life and human interactions and that sort of thing, that's where we really have to listen to patients about what they care about. So I think all the above, but I think at different paces.
Well, that's great. I mean, to certainly hear that progress is happening in all fronts, I think that's something that should inspire everyone and make them feel good for any loved ones you might be concerned about down the road a bit. And you kind of answered this next question a little before, but I'll ask it again, since it helps support what the foundation is doing. But just in terms of research in general, for everyone who's on here, and again, we had about 100 people register for this, I'll probably go back and send everyone who's on and who did register. I think many of you know that the foundation does go to Capitol Hill every year, advocating for greater research funding through NIH, particularly NINDS, with a legislation called Ellies Law. And since we went in March, we've had, I think, 9 new people in the House side sign up. So we have, I think, 50 some people there, and we're trying to get to 100 to really get this legislation through. And the Senate is slow, but also starting to build up. So I think the momentum is there. And I think if all of you get this link, that you can just, through your computer, encourage your representatives to support this research funding, so people like Dr. Jackson, who are working in really important things, can get more funding than the foundation is able to provide. I think it's just critical. So that's something I will do when we have the recording and I send it out to everybody. I am going to send that link and just ask you all to please sign up and ask your members of the House and the Senate to support Ellies Law. It's not a big ask. It's $10 million additional from what they are currently funding, which is only a little over $2 per person affected each year. So it is critical. But again, Dr. Jackson, if you can just speak to the importance of the funding the foundation and other foundations are able to provide.
Yeah, I mean, for this type of research, it's absolutely critical. I mean, there's no doubt about that. I think I alluded to this a little bit before, but I think brain aneurysms, sort of fall. If there's a theme here, it's this sort of middle ground, right? Where, it's not, so if you take a orphan disease, you take a muscular dystrophy, gene therapy sort of thing, there's a lot of, in part because of good advocacy and that sort of thing, but also sort of because of some of the incentives that do exist. A lot of people, a lot of, pharmaceutical companies, a lot, grants are available, it sort of gets attention. If you look at something like heart attack, where it's a huge killer of Americans, again, lots of funding, lots of things available. Brain aneurysms, it's something near and dear to those of us who take care of those patients and for people who are affected, but it doesn't capture sort of the public in the same way. You don't have, the same, for all those reasons, I think. And, I think it's also a little bit harder for people to grasp, to be honest. I mean, the people in this room, virtual room, are very well acquainted to this. But if you stop somebody on the street and you start talking about, aneurysms, then you have to talk about rupture, and then you have to talk about delayed cerebral ischemia and vasospasm pretty quick. People kind of glaze over. So I think all those things make it kind of hard. So what's important is, when you have a topic that I think is a little bit harder for the public to grasp and that is not so prevalent that, at a societal level, it's sort of perceived to be a major public health issue. I would argue that, but the numbers are what they are. You have to come with data, right? So you can't just come with an idea. You have to come with data. And the only way to get data is to have some support, to have somebody sort of listen early on, and provide support. And that exactly is what the Brain Aneurysm Foundation, feels and has done. And it's actually, I think, I'm involved with several, so I have several foundation grants for various things. And, I will say the Brain Aneurysm Foundation, in my experience, is sort of the most personal and sort of the most passionate, actually, if you will. I mean, to get to meet the people, who raise the money, hear their story. I mean, all of these things are really, really unique. And, and the people who are funded by the Brain Aneurysm Foundation tend to also be a fairly close, tight-knit group. I mean, so it's not just the money, it's the connections, it's the, and again, and it's a small world, and that we all sort of keep mixing ideas. And so it's the money, but it's also the forum to do that. No, I mean, it's absolutely critical. I mean, without that, just new ideas just wouldn't ever have legs. I mean, you have to have that initial funding to come with data. And then once you have the data, you can get the attention of whether it be pharmaceutical companies or whether it be the NIH or whatever. But, you have to start with the data, and the Brain Aneurysm Foundation is critical for that.
Okay, thanks, Dr. Jackson. So now we're going to go to some questions that people had submitted before, and there might be a few in the chat. And some of them might touch a little bit on what you have talked about, but I think just for additional clarification and make sure they understand it, and I'm addressing their questions. Are there any known methods to reduce vasospasm in the first week after surgery?
Yeah, nimodipine, interestingly, so I mean, just a little bit to be precise. So it's interesting, and nimodipine is the only drug approved, again, to improve outcomes in vasospasm, delayed cerebral ischemia. But it's interesting, it doesn't actually affect vasospasm as we measure the vessels. It affects the outcomes, right? So, and that's the only drug. So most of it really is supportive, and then in sort of an emergency situation, if once somebody has developed vasospasm, to go in and try to open those vessels. But that, of course, is invasive and carries significant risks, and you're kind of on your heels at that point. So I think what's really needed and what we're working on is something earlier than that, something that's truly preventative. And I think that would be a game changer.
Excellent. Another question, what are the most reliable factors for predicting cerebral vasospasm?
Yeah, so it's basically how much blood is in around the brain, and then some patient factors, like I said, like younger age, female sex, that sort of thing. But you can't, and in time, of course, like I said, self-limited 3 to 14 days, and it tends to be most prevalent around day 7 or 8. But you really can't predict. I mean, I have seen patients that I thought for sure would go into vasospasm who didn't. And I've seen people who I thought for sure wouldn't who did. So, we end up just sort of having to play defense and treat everybody the same and react promptly.
Okay, thank you. Another question, can you visualize vasospasm with CT perfusion, and how do you differentiate it from other conditions?
Yeah, no, that's a really good question. So yes, CTA and CTP, CT perfusion, CT angiography can visualize vasospasm. Conventional angiography, is sort of the gold standard, but you can see that. And no, it's a good question. So, you're basically looking for a series of things together, right? So you could have a stroke just from the aneurysm rupturing, right? Like you could have an area of ischemic brain that on CT perfusion, looks like a stroke. But if the vessels look completely normal and wide open, then it's probably not really from vasospasm, right? Or the opposite can be true. You can have, a patient who has a CTA that looks like vasospasm, but because of those other pathways, they're getting blood to the brain and they don't actually have, an ischemic area. So it is tricky, but it's a series of things together. Any single variable could be due to something else, but when you see the pattern, that's vasospasm and DCI.
Thank you. Can a EEG detect vasospasm?
No, it can't. Not specifically, anyway. No. So you do see disruption in the brain waves, and there's some people certainly studying how vasospasm and delayed cerebral ischemia affect how neurons fire, the electrical activity, which is what an EEG picks up. But no, there's nothing clinically available with an EEG to detect vasospasm specifically.
Okay. Another question. My wife suffered a level 5 plus brain aneurysm. She had severe vasospasm. She was given Verapamil,
Right. Uh-huh.
but they no longer use it. She was in a coma, hypothermia, coded twice, but lived to be a miracle. What do you think of this?
I mean, first of all, I'm sorry you went through that, but it's also remarkable because what you're describing is, as harrowing as it gets. So verapamil is still used intra-arterially to open the vessel. So I mentioned with an angiogram, you can go and give it, very close range. It's one of the drugs that can be given for that. It works well if you have sort of a specific artery affected, right? So if you only have like one area, you can imagine, you can park the catheter right there and you give the drug, and that works pretty well. The problem is it doesn't always last. So sometimes people are going back to the angiography suite, every day, through the entire vasospasm period with, a new angiogram, all the risks that come with that, discomfort that comes with that. And the other thing is that when people have, very severe global vasospasm, so involving, multiple different areas, different arteries, they can't always get to all of that, or it doesn't work as well. So those are the challenges there.
Okay, thank you. Another question, is there any data on if an aneurysm is in a certain place or shape increases the risk of vasospasm, and why do they occur 7 to 14 days after surgery? Is there any way to treat the patient right after surgery to lessen the chance of occurrence?
That's a great, both of those are really great questions. So in terms of location, not reliably. Like everybody who does this, thinks, for example, that maybe ACOMs are a little bit worse, and we sort of have just reasons that we think that, but not in a way that we can mechanistically, put the data down and then use for guidelines, which is what we really care about right now. Maybe we as clinicians watch those patients a little more closely, but there's nothing that we can use, with any sort of statistical reliability to do that. Your question about stopping it is exactly kind of where my mind is on this, and I think that's what's most needed. So again, you give nimodipine from the beginning, which is probably mostly just neuroprotective, but you can't actually stop it from happening. But that is also related to what you're asking about the timing. So we think that the timing of the delay is because you have this event that happens in the brain. It causes an inflammatory response all the way down to, in animals, you can actually see a difference in the bone marrow. So you can see inflammation start in the bone marrow from what happens in the brain. But it takes a day or so for those signals to start. And then those immune cells, the inflammatory cells, the white blood cells, have to leave the bone marrow, travel through the blood, and then get into the brain to cause the vasospasm. So we think that timing is all related to that response, that peripheral immune response. You can think of it, I mean, I hate to bring this up, we're coming to summer and COVID is going away and all that good stuff. You can think about when people got very sick from COVID, it usually was not sort of the initial event where people got severely ill or life-threateningly ill. It was days after, because of the inflammatory response that they were having to the virus, not just the virus itself. So inflammation kind of works that way. Or you can even think about, if you get a shot in your arm, it doesn't really hurt so bad immediately when you get your flu shot. It's a day or two after that it hurts. It's the time that it takes for the immune response to get generated. And most of the time it's trying to do something good for us, but occasionally it's doing something counterproductive. And in this case, it's largely doing something counterproductive. But we think if we can intercept that before the effects, that maybe we can, in fact, come up with an abortive therapy.
Thank you. Is there any data on whether an aneurysm is clipped versus a web device used that it would either cause more or less vasospasms?
Oh, that's a good question. Yeah, so you can get vasospasm either way. Certainly in patients who are already in vasospasm and those vessels are reactive, we often try to avoid surgery for those patients. You know, we'll even in a patient that might not be the best candidate for coiling because of the shape of the aneurysm or the location, sometimes, you know, we'll treat it with coiling, even knowing that more treatment would be required later to avoid manipulating those vessels. Because what we do often see, you know, if you operate on a patient who's in vasospasm, is that, you know, and you can see it literally under the microscope, is that those vessels start to pinch off from the manipulation. So there's nothing as far as what's more likely to put you into vasospasm if you're not already there. But if you are there, we try to avoid surgery in favor of endovascular treatment.
Great, thank you. Can a vasospasm be present with an unruptured aneurysm?
Well, that's a good question. So vasospasm can happen to people actually in other conditions. So it's very, very rare, you know, but there are conditions where, you know, you can have vasospasm even in the absence of blood in the brain. Now, as far as we know, having an aneurysm doesn't necessarily predispose you to that. We think those are two separate things, but it's actually a fascinating question that I hadn't quite thought of as to whether or not, you know, there's some common pathway of the inflammation that would cause an aneurysm also causing, you know, people to be more susceptible to spontaneous vasospasm without it rupturing. So interesting. Sounds like a good grant for the Brain Aneurysm Foundation.
Yeah. And then another question, is there a correlation between susceptibility to vasospasm and autoimmune disease?
Also a good question. That's very hard to study. So nobody has pinpointed that. But what I would say, because, you know, you'd have to have patients with sort of the same autoimmune disease on the same treatment, also rupturing aneurysms. I mean, you know, that gets to be a pretty hard study to do just based on pure numbers. So the answer to that directly is no. Indirectly, something I've alluded to a couple times is that, you know, the patient population most likely to get vasospasm is young women. And that also turns out to be a population that is, you know, most likely to get autoimmune disease. And I think the underlying commonality there is that, you know, they have very active immune systems. You know, young women tend to be, among people, have the most active immune systems. And so, you know, I think both are obviously immunologically related, but not necessarily like if you have autoimmune disease, you're more likely to get vasospasm, at least as far as we know.
Okay. Another question, how often does a stent that does not deploy correctly possibly lead to a TIA, or is a TIA in any way related to a vasospasm?
So both are ischemic events. So in other words, both are sort of strokes or precursors to strokes. You know, a TIA is basically an almost stroke. You know, basically there's a part of the brain that's not getting enough blood flow, and then the rest of the arteries manage to make up for it sort of at the last minute. You can think of it that way. So there's no residual effects. And that's basically what a TIA is. You know, a stent that doesn't deploy correctly, you know, certainly can cause strokes, you know, or TIAs, absolutely, because it narrows the vessel or causes, you know, platelets to stick to it, and then that causes clots. So yeah, that certainly can be.
All right. I think those are the most pointed questions related to the topic here. So I think we'll wrap things up. And I just want to say a big thank you, Dr. Jackson, for, you know, taking the time to share this information with us. It definitely matters. And again, this is going to be recorded. We share it out and we'll go to our YouTube page. But any closing remarks from you, Dr. Jackson?
No, just thanks again, you know, well, first of all, for the support of the foundation, the continued support of the foundation and putting together things like this, and, you know, for the opportunity to talk to everybody who signed on today. You know, if there are any additional questions or anything, you know, that I can answer, you know, feel free to reach out. You know, Christine has my email, you know, and I'm happy to chat further.
All right. Well, thank you, everybody, for being on this month. We'll see you next month with a topic to be determined. So enjoy the rest of your week. Thank you. Thanks again, Dr. Jackson.